In this video, we are going to study about the estimation of hemoglobin with the help of Sahli's hemoglobinometer set manually. So the requirements for estimation of hemoglobin is Sahli's hemoglobinometer set, which contain first hemoglobinometer tube, which has two markings. First is in percentage value and second is in gram per 100 ml. Usually the hemoglobin is reported as grams of hemoglobin per 100 ml. Then there is hemoglobinometer pipette, which has 20 microliter marking up to which we have to fill the blood. Then the comparator box, which has color strips to compare. Then N/10 HCl. Here the N means the normality. Remember that we cannot use normal HCl or any strong acid or alkali because it will cause the destruction of proteins present in the hemoglobin and hemoglobin value will alter. Then stirrer, which is used for mixing purpose, and dropper. Then the other requirement is blood sample and the tissue paper for wiping off the extra blood. Before doing the whole process, firstly let's see the principle. The amount of hemoglobin can be estimated by conversion of known volume of blood into acid hematin. So the acid hematin is formed when the hemoglobin is reacted with N/10 HCl. So the brown colored which is developed is matched with the colored glass standard of the comparator box. Let's see the whole process. So firstly, with the help of dropper, fill N/10 HCl in hemoglobinometer tube up to its lowest marking, that is 2 mark on gram by 100 ml. Then put the hemoglobinometer tube inside the comparator box. Then mix the sample. Then with the help of hemoglobinometer pipette, suck the blood up to 20 microliter marking. Then wipe out the blood sticking on the hemoglobinometer pipette before transferring the blood into the tube. Then immediately draw the blood into the hemoglobinometer tube containing N/10 HCl up to the 2 marking. Then mix the content thoroughly and put hemoglobinometer tube in the comparator box. Then wait for 10 minutes. During this time, hemoglobin is converted to acid hematin, which looks brown in color. Then after 10 minutes, add N/10 HCl or distilled water drop by drop till the color of the solution is same as that of comparator box color strip. Mix it with the help of the stirrer. For dilution, we can use both distilled water or N/10 HCl. But remember that we cannot use the tap water for dilution because it is not pure and contain microorganisms. After exact matching of the color, take out the stirrer from the solution and read the meniscus of the fluid in a graded tube from gram scales. The lower meniscus need to be readed. Lastly, I want to tell you the mistakes which should be avoided while doing this process. Firstly, all the apparatus must be cleaned properly. Matching of colors should be done after lifting the stirrer from the solution. Then wipe out the blood sticking on the hemoglobin pipette before transferring the blood into the tube and try to compare the color exactly. Then let's see the normal hemoglobin values of different animals, which is in gram per dl. Firstly, for horse 11.5 to 16 gram per dl. Then for cow 8 to 15 gram per dl. Then buffaloes 9 to 13.5 gram per dl. Then for sheep 9 to 15 gram per dl. Then for goat 8 to 12 gram per dl. Then for pigs 10 to 16 gram per dl. Then for dogs 12 to 18 gram per dl. Then for cats 10 to 15 gram per dl. Then for chickens 8 to 13 gram per dl. Here we ends the topic estimation of hemoglobin.